Hi, everyone. Welcome to Neuroscientists Talk Shop, UTSA. I start to say UCLA because our guest today is from UCLA. UTSA's Neuroscience Research Podcast. Today is March 3rd, 2022, and we're chatting with Dr. Bennett Novitch, Professor of Neurobiology and the Broad Center of Regenerative Medicine and Stem Cell Research at the David Geffen School of Medicine at UCLA.
Yeah, say that several times in a row.
I've actually practiced it a couple of times, and that's as good as I can do. So hi, Ben.
Hi, how are you?
Welcome. So Ben's interests are in brain and spinal cord development, specification of cell types, and circuit formation. And that's my read of his interests, not his. He can correct me if I'm wrong. He uses human cell-based models to understand the developmental origin of circuit dysfunction in brain disease. And joining us is our own local expert on these very same issues, Jenny Shea. Hi, Jenny.
Hi, everyone.
And I'm your host, Charlie Wilson. So Ben, you build designer cortical circuits from scratch. Using pluripotent stem cells, evaluate their cell types, connections, physiological function. Sometimes you make them out of cells from patients that have a genetic disease, like Rett syndrome, and you're able to identify aspects of the disease functional phenotype in those circuits. It sounds so fantastic, but it's true.
Yeah.
And to me, the most mysterious thing is all the different cells that you start with the same cells, you treat them all the same, and you can end up with lots of different cell types. For example, all the different cortical interneuron types that you get from the ganglionic immanence. Now, of course, that happens in normal development too, but there could be a million other things happening in normal development, but you know what you've done to these cells. So that's amazing. So to get us started, could you in broad strokes just sort of summarize what you've learned about what's required for a set of cells to differentiate into all the different neuron types that are found in the cortex, wire themselves together, and to make a working circuit?
Well, I mean, I'd say that, really the master is nature, because what we're doing is essentially just recapitulating what happens in actual real human brain biology. So what we're doing is the things we learned was that the way you start off with your cells, like with the starting place of the cells in terms of the state of their pluripotency, which means basically their developmental potential to give rise to different cell types in the body, that can matter. So you could basically get good or bad outcomes depending on where the cells start from. And then what we're doing is we're basically steering the cells to first form neural tissue, and then the neural tissue has a lot of self-organizing principles, and the cells communicate with one another, and that's part of the process by which different cell types are born, because cells kind of know that they have a place, and they can communicate with their neighbors to say that you do this and I'll do that, and that's one of the ways in which you end up with the diversity of cell types. I mean, there's some tricks that we've learned along the way, like for example, when we make an organoid, we found that there's a sort of a sweet spot in terms of the numbers of cells that you have to put together to form the initial organoid, and we have to grow it in certain media conditions that allows for that ball of cells to grow, and for the cells to epithelialize, which is to basically make the connections that they need to make with one another to make a vesicle structure, and then to expand and differentiate and so on. So a lot of this is just sort of encouraging all the inherent mechanisms that the brain tissue already knows how to operate.
So when a cell is born, when it undergoes the final division in the medial ganglionic immanence, what determines whether it's going to be a VIP cell or a somatostatin cell? I mean, is it not determined yet? Maybe, and it's going to talk. It's going to say, hey, how many VIP neurons are there out there? Well, not very many. Maybe I'll be one of those. Does it happen late like that, or is it?
Yeah, you've hit upon one of the more controversial questions, which is presumably why you picked that one. So to begin with, I mean, when we make the cortical organoids, they don't have a capacity to make many inhibitory interneurons, at least not with our particular organoids. But we can direct them to make basal ganglia and to make the progenitors for the inhibitory interneurons by treating them with patterning factors that we know operate in embryogenesis to ventralize the cells. So something called sonic hedgehog is the pathway that's used to ventralize, and we can use chemical agonists of that to make the organoids turn into the progenitors for the inhibitory interneurons. What happens then is still quite mysterious. There's a lot of studies that are going on. I'd say that the most advanced are actually right now in the area of rodents, mouse studies in particular, trying to understand where does the diversity come from. And so there are at least two models that are generally thought of. One is a positional model, which is one that is kind of near to my heart because I used to work in the developing spinal cord, and one of the things we were interested in is there are different types of spinal neurons, motor neurons, and different types of interneurons, and they all form in a kind of a dorsal ventral register along the spinal cord, and that all is related to signaling gradients that exist along the dorsal ventral axis of the spinal cord, of which sonic hedgehog is one. And so there's a clear register of progenitor groups which give rise, they're dedicated to the formation of individual classes of neurons. And so when we get to the basal ganglia, I think in broad strokes that is true, that there are like more ventral and less ventral progenitor populations that would be more likely to give rise to, say the cholinergic neurons or other, somatostatin neurons and all that. So this is like MGE, LGE, and so on. But then there's also potentially diversity within each one of those progenitor pools. And so there's a lot of interest in whether or not there are like temporal mechanisms by which one neuron is produced at an earlier time, and then other types of neurons are produced at later times. There's evidence for kind of like post-mitotic changes in some cases, I think, for neurotransmitter phenotypes. And you can imagine there are other mechanisms by which you could contribute further to diversity. So it's a little bit of everything, I would say.
And one of the things that struck me in your talk today was that there's something like the ratio of these excitatory to inhibitory cells seems to be regulated somehow. And like you mentioned, something like for every 2 or 3 excitatory neurons, there's 1 inhibitory neuron. And it's amazing how that can be coordinated so precisely, even in an in vitro system.
Yeah, no, this is actually like something that amazes me as well. So what you're hitting upon is that when one looks at an intact brain in many species, that there's this magic number of about 20 to 30% or so of the cells are inhibitory, where the rest are excitatory. And how do we achieve that balance? And in our model, we're taking these two different types of organoids, one that's predominantly excitatory because it's coming from a cortical developmental pathway, and then the other being a largely inhibitory because it's coming from the ganglionic eminence developmental pathway. And we stick these two pieces together, and the interneurons can migrate from the ganglionic eminence into the cortical organoid, and then we see this population of mixture of so. So how can we account for the precision? I mean, precision is probably an overstatement, but this kind of range of density of the inhibitory cells, and we don't really know. But it's something that is. I had a conversation with another investigator from UCSF earlier this week, who's been doing experiments of transplanting the MGE, the medial ganglionic eminence progenitors, from one animal into another, and trying to rescue developmental phenotypes and epilepsy as well. And he also has these observations that they've never seen overpopulation. So meaning that there is some sort of a regulative process by which the balance is maintained. And so I don't have here's the answer. But one of the ways that I've been thinking about this so far, and I may change, is that there clearly are things that are related to cell numbers in terms of cell survival. So I think that there's clearly in development, we know that there are mechanisms for assuring the appropriate number of cells matches the need for the circuits by what are called neurotrophic mechanisms, by which the cells that are excessive, if they don't make connections properly, they lack a reward signal that benefits their survival. And so if you have excessive numbers of cells, that won't participate in the circuit, they will die off. And we have seen evidence of that. If you make some ganglionic eminence organoids and you look for certain populations of neurons, if you just left it as just a ganglionic eminence organoid, we don't see many of them at the end of the experiment. But when we do the fusions, we see more of them. So I think that there's something about those cells being in the right environment that allows them to participate in the circuit and thus survive. And so I think that that's one of the primary mechanisms by which you could potentially account for why the balance is there. The other is whether or not there is just some sort of like limited availability for synaptic connections on the inhibitory side, synaptic connections from inhibitory cells. If I'm kind of going to make a flub of this, but that they're more constrained to the soma and like less the branches, the more distal branches, and so they have like more limited real estate with which they were likely to be forming connections, and so that may also sort of like limit just the possibility of the connectivity that they could be making.
So it's not that migration that is regulated per se, it's the survival of the migrating cells, you think?
Well, I mean, migration is, so those cells, the inhibitory neurons do migrate, obviously. And so you could imagine, and this has been well documented by many other people's studies, that there are developmental defects that affect migration. And you can definitely see like deficits in the interneuron populations as a consequence of that. But in our case, I mean, the mutations that we've been studying don't seem to impact migration, so thus we wouldn't see it in our system. But I think that migrations could definitely have an impact, but just not in our particular experiment.
So what you just said about the ganglionic eminence organoid not surviving by itself very well makes me wonder about whether the cortical organoid is actually growing axons into the ganglionic eminence one. The ganglionic eminence gives rise to one of the biggest targets of cortical axons in the whole brain, and so why wouldn't the cortical organoid innervate it? Do you know whether it does or not?
We haven't really gone looking for it, but it doesn't surprise me that there is something going on, because we have done these experiments where and they were designed principally to track the migration of the ganglionic eminence neurons going into the cortex. So we would label the ganglionic eminence organoid with a tracer, and then watch that tracer move into the cortex. But we did as control, we would label the cortex and see whether or not any of that tracer would go over into the ganglionic eminence. And what we saw there was we didn't see a whole lot of cell soma, certainly not at the cell bodies of the neurons, which would be indicating like a mass migration. But if you wait long enough over time, you can start to see stuff kind of going into the ganglionic eminence. And what I never really, went looking for was whether or not those were more neuronal processes rather than actual like cells themselves migrating, whether or not this was the axons or other processes that were,
So you show these like really striking phenotypes, like in terms of this like hypersynchrony in the fusions of looking at mutant combinations, these MECP2 mutants compared to the control. And my understanding is that these fusions, these fused organoids are sort of development, if sort of more representing embryonic developmental stages. So, but do you think that in the actual patients that they would be having these same circuit types of abnormalities that early, like maybe in utero even, because it seems like in real life the patients, they're developmental but not that early, or is it possible we're just missing that because it's so inaccessible?
Yeah, I think there's several several answers or responses to your question. So one is, I think for sure we're missing things, because I think that they're, like we don't often like go looking for these things until they become a problem, and that's usually not manifest until a child is born. Mm. But in the case, there are cases of, I think, some genetic epilepsies where you can actually see in utero hypersynchronies and such it has been done and has been observed. Another thing that may confound our studies a little bit, or at least like trying to relate our studies to the human patients for Rett syndrome, is that the nature of the experimental design that we used. So in our experimental design, this is kind of like version 1 of the experiment, was to take Rett syndrome patients who themselves are actually heterozygous. So they basically have a gene mutation in the MECP2 gene, but it's only on one allele. And so the patients are actually mosaic, where only half of their cells, or on average, we'd say, are actually mutant. And so what we don't know is whether or not that has like some way of sort of like normalizing the activities. And what we're actually creating, because of just the experimental design, is that we're making organoids that have no MECP2 in any of the cells. And so whether or not that is, that we're just seeing like a very, very severe version of what the patient would be having. That said, we are actually currently working on version 2, which is to actually make mosaic organoids, where we can basically start with the stem cells that are coming from a patient, and we can basically dial in the ratio of wild type and mutant cells together to create organoids of different mosaicism. And then we're looking to ask, how does that manifest in terms of the neural oscillation activities? Thus far, what we've seen is that they seem to have an intermediate phenotype, meaning that they seem to have less hypersynchronies, but they actually still show some defects in the oscillatory rhythms. And so we're excited about this because we feel like this might be a better model for the actual patient disorder. And at the same time, it also allows us to ask interesting questions about the way that mosaicism for a defect, how does that manifest at the network level? So in other words, how many bad apples in a circuit does it take to bring the whole circuit down? So that's one of the questions that we're trying to ask.
I think I should say that the interest in this mutation and hypersynchrony that results is because this mutation is associated with epilepsy in humans. So hypersynchrony and changes in oscillation are like, I don't know how much like, but a little bit like epileptic activity in the cortex.
Thank you for rescuing me, Charlie. Yes, I mean, it's very true that, I mean, the things that we see in terms of like the epileptiform activities that we can see in these organoids, and I know that this is it's a very reduced model, but it has striking similarity. I mean, Rett syndrome patients, I think it's like over 80% or something like that, have seizure episodes, and so it's a very high, like high incident comorbidity. So I think it's the fact that we can see them at all, I think, is quite relevant. We like to believe.
And the electrical activity that's happening in the what's the right word? We shouldn't say wild type, but we say that when we're talking about mice or something. But in the normal,
in the native,
native, yeah. There you go. I like that. The organoid is also interesting. It's desynchronized. It looks like it could consist of cells bursting at a fairly low rate and could somehow play a part in network organization and synaptic arrangements. It does. So I'm wondering about synaptogenesis along the time scale of the things that you're studying. When are synapses forming in the organoid? I guess in the organoid is what's interesting because it doesn't have to exactly correspond to anything that happens in the human development in time.
I mean, the thing that we've been very limited by is the resolution of synaptogenesis. This is something has been dogging us for a while, and we've been making progress towards it, but it's still something that's quite difficult because the synapses are small. It's not so easy to image with light microscopy. But we can do it, and we haven't been able to track some the densities of different types of either excitatory, inhibitory synapses, and we see that they form exactly when you would expect them to form. Soon after the neurons are there, you can start to see the synaptic densities forming, and we can follow those things out over time. But we don't have a way to look at, I think, in a dynamic way, a dynamic manner to actually look at synaptic remodeling in a real-time way. These are things that may be technically possible to do. We just haven't done them yet.
So I'm just sort of wondering if some of the answer to my original question about cell types and circuit diversity could be explained by electrical interactions between the cells, ordinary synaptic interactions like the kind that synaptic plasticity and the kinds of things that control network activity under normal circumstances. So if the cell types are all completely determined before the electrical activity happens, then that couldn't be participating in it. But if they're not, then it could. Is it
Well, I mean, I think that one part of it would be cell determination in terms of like the different types of cells, but the densities, the ratios of those cells. Like even though we say that like there's a set like ratio of inhibitory interneurons, I mean like the composition of like do you have more of certain classes over others, that could have profound impact on the way that the circuit is going to function. And then on top of that, you could consider just the activity of a given series of neurons in terms of like are they providing a really strong, say, inhibitory drive, that, and like who are they connecting to? That's going to change the oscillatory patterns as well. So you could have some that like completely just like squash all oscillations, and other ones that squash the inhibitors of those oscillations. And so depending on who's driving whom, you could see like changes in just the overall tone.
So when things work out and the oscillations happen the way they're supposed to, that means that something went right in determining the different kinds of interneurons, right? Some interneurons end out on the distal dendrites, some on the soma. You see all the diversity of interneurons in the organoid that is normally seen in the cortex.
Well, I'd say we see a lot. I would never want to say we see all, but we see, in my mind, a pretty remarkable diversity.
Well, I think your work is probably one of the, I would say, gold standards, because I've noticed so many like different classes of mature interneurons. Depending on the study and the different protocols people are using, people are finding very different things. And but interneuron differentiation, maturation has always been like very tough for IPS cells. Would you agree?
Yeah, absolutely true. But that's a lot of that comes from the 2D culture, which was where most of us were for the past decade. And so, I think that the issue is that in most cases, people were doing these things with isolated cultures. And if you have a monoculture of inhibitory neurons, they don't seem to mature. And, they only get maturation when you transplant them into an animal, or we would like to believe that when you put them into a context with the organoid, where they're in contact with the cortical neurons or other targets that they might be interacting with. And so I think that there's something about the feedback that neurons give to one another that drives their functional maturation.
So part of what's happening is just working out the technology to make more and more elaborate versions of these circuits, and I wonder how far it'll go. So, I'm anxious for you to see the cortex hook up to the basal ganglia, but I'm also thinking about the thalamus. A lot of times people say, thalamic innervation of the cortex is partly determining the organization of cortical areas. And there's no thalamus there yet, but I suppose the thalamus could be sort of around the corner. Do we know how to make a thalamus?
Yeah, so my lab hasn't done this, but other labs have. And so people are already on this problem of trying to look at like thalamocortical organoid connections, and other labs have actually gone to looking for like outflow pathways for neural activities coming from the cortical organoids. So, for example, at least two groups have taken cortical organoids and put them together with either spinal cord and muscle creps coming from a mouse, or created spinal cord organoids, or like mixtures of spinal cord neurons along with muscle cells, and actually shown that you can get corticospinal connections forming from the organoid to the spinal cord cells, and then the motor neurons from the spinal cord cells, making connections to the muscle, such that you could elicit like a signal starting from the cortex and elicit a muscle contraction. So this has been done already. So
I'm surprised that wasn't you, since you're the spinal cord. I'm
surprised it wasn't too, especially since part of my lab has worked on the spinal cord to muscle side of things. And anyways. But I applaud the other labs that have done these experiments, because I think they really show like again, like just the possibilities of being able to sort of assemble these different pieces of the brain and spinal cord. And so I think it kind of like gives me a lot of hope that we might be able to use these in, like, as you said, like to try to be able to study the interactions of one region of the brain on the other for development. I think it's going to be really powerful.
And we should mention something about the medical applications and especially personalized medicine, because I think that drives a lot of this work. I mean, I'm super interested in making a whole brain that would work, but
I mean, you mentioned in your talk, testing different pharmacological agents. Another possibility for therapeutic strategy is gene therapy. And I think a lot of people are trying to work on Rett syndrome, right? There's a lot of companies probably working in this area. But I think one of the questions is, do you deliver MECP2 everywhere to every cell type? And when is there a critical or a therapeutic window? And perhaps the organoid system could be part of that, just to answer some of those basic questions. What do you think?
Yeah, I mean, I think that the organoids, I think that just as an extension of IPS research, is a game changer for the field of therapeutic discovery, because just the ability to be able to make human brain tissue in a scalable manner is, I think, huge. And the more that we are able to make things with network-like functions, where we think the disease manifests, is going to make it possible to really create models for a whole variety of different types of disorders. And if you can reduce the scale of where you think the defect lies, this then puts it into at least the arena of being, if it's not usable for high throughput screening, it's certainly for like the lower throughput screening and preclinical validation of a drug being useful for a given model. I think that the question that you raise about like when would you need to intervene is a really, really tough one. And so far I'd say like the jury is very much out. I'm not really going to be relating much here about organoid research, but there's been a lot of research that's going into animal studies where models for different disorders have been created. I actually saw a really wonderful talk on Angelman syndrome recently, and some therapeutic approaches there to try to restore, like using a gene therapy approach. And some of I think the findings there suggest that you really do need to be able to put the gene in during a critical period of early development for it to be functional. And so this will pose a significant challenge. Like that we have to think about prenatal diagnosis and prenatal intervention potentially. And so this is definitely going to be a hard puzzle to solve. On the other hand, the way that we're also trying to think about what we're finding from the neural oscillation side of things, that we know that if you have, for example, and I think that this is very connected to epilepsy research, that if you're having a seizure all the time, the other functions of the brain that depend on oscillatory activities can't happen. And so even if we could just use the organoid platform as a way of identifying and testing various therapies that could intervene with seizure behavior, that could have a knock-on effect of really helping overall brain functions. And we also know, it's long been known that there's a high comorbidity between seizure disorders and autism spectrum disorder, for example. Mm-hmm. So I think that if we can solve one of those problems, I mean, maybe starting with the epilepsy side of it, the seizure side of it, we may actually have some benefits on the other as well.
Yeah, because my understanding is in these developmental and epileptic encephalopathies, or these DEE syndromes, mm-hmm. that it's the seizures they're sort of developmentally on track until the seizures start, and then they regress. So to your point.
I mean, and that, getting back to your question about the Rett syndrome model, the fact that we see seizures from the outset here, kind of potentially is why the whole system is dragged down, because it's potentially grown up with having that effect. But yeah.
And especially, I think if the electrical activity is part of the normal network formation process, then abnormal electrical activity ensures an abnormal network formation process. And
getting back to your original point.
So in the best scenario, these things become patient by patient. I hear that all the time. So that's, in this case, the Rett syndrome, it's not really to that point, I guess, because we don't expect that it's one mutation. We don't expect that every patient is going to be different from every other patient. But in some kinds of disease, it may be something really patient-specific is required. Is that part of the promise for the future for organoids?
Well, I'd say that, I mean, it depends on the disorder. I mean, so even with Rett syndrome, this is something that we're actually setting out to test, is that we know that there are different gene mutations. They're all the same gene mutation. They're all mutations in the same gene, MECP2, but there are different, ways in which the gene has been disrupted and the proteins that are produced that seem to have either higher or lower clinical severity. So one of the things that we actually wanted to test to try to get to some of these questions is, can we actually see gradation in the pathology, or whatever we want to call the abnormalities in neural oscillations or other aspects of neural functions in organoids that are made from patients with these different types of mutations. Because I think that that's something that's important for us to know, is like, how sensitive is the system to being able to distinguish more or less, like something that's going to be truly like just debilitating from something that's more innocuous. So, because otherwise we just don't really know, like when you say like we want to like be able to try to match this to patients, we need to know a little bit more about just the how effective the organoid model is going to be for different disorders. And so we need to know the range of utility that it has. And I mean, the hope would be that, when we talk about different diseases, I mean, we tend to, I think, gravitate towards single gene mutations, because as experimentalists, we know that they're far easier to work with than, say, sporadic disease. And sporadic disease is sort of like the great unknown, that we really don't know how that's going to manifest in these sort of models. But that said, I think that if we can create organoid models from sporadic disease, we might be able to then look at the organoid itself using methods like single cell transcriptomics, and start to identify clues as to which cell populations look abnormal or activities that seem aberrant, and then sort of develop some new hypotheses about what the causes of those sporadic diseases are. But it's still, it's a harder path to walk.
Wow, that seems like a encouraging prospect for the future. So maybe we should stop there while we're ahead. Thank you very much, Bennett Novitch, for joining us.
Thank you for inviting me for this conversation.
And Ginny, this has been Neuroscientists Talk Shop.